Welcome to this webinar. During this presentation, we will give you some insight about the Biofilm Expert project, including the background of the project, some results, and conclusions. So, first of all, let's talk about some numbers. Globally, it is estimated that 1/3 of the foodstuff that are produced in the world are wasted. So in total, it's 1.3 billion tons per year. In Europe, it's about 88 million tons. It's on average 280 kilograms per inhabitant. And the global cost is about 143 billions of euros. So it's a huge amount. And in order to act on those wasted food, Relco decided to initiate a Biofilm Expert research project in the objective to reduce this huge amount of wasted food. The objective of this program is to evaluate the capacity of enzymatic cleaning to act on the microbial flora of equipments and finished products. And the idea is to compare with standard chemicals or conventional cleaning process. A few partners were in this project, generalized partner, but also University of Liège in Belgium and Relco. It's a 2-year study, and we tested our solution in different food application. It can be ready-to-eat food. It can be collective kitchens, meat processing plant, cheese factory, and butchery. In total, it's more than 1,000 samples that have been collected from the field. And this project has been financed by the Walloon region. So why this study is unique? It's many reasons. First one is we make the comparison between enzymatic cleaning and traditional cleaning to see if there is a difference between those two ways of cleaning. It includes many samples all over a long period in different field application and different cleaning protocols. Also, we are introducing a new technology or alternative technology, the metagenetic, in order to analyze germs that are present in open surface, in closed system, but also in the finished product. And this study gives provides a solution to improve microbiological quality of food products and in order to see if there is an impact on the shelf life of the finished product. If you want to get all the information about this study has been published in Frontiers Microbiology magazine, but also you can find some additional information in our website. Today in this presentation, I will introduce one case. It's a company that is producing Italian dishes. And in this factory, we have, as I said, tested different cleaning treatments. You will always see in this presentation that dark blue is conventional cleaning. Dark green is reinforced enzymatic cleaning, and light green is routine enzymatic cleaning. And you will get some results about the impact of those three different cleaning procedures. Obviously, all factors during the project remain constant. We don't want to change too much parameter. In this factory, it's about 189 samples that have been analyzed, including some surface samples, but also some samples collected from the finished food at D0, so at the end of the production, also at the end of the shelf life, but also 7 days after the end of the shelf life. And those samples have been analyzed via classic microbiology, but also metagenetic. In order to see if there are differences between those different cleaning procedures, we are using some statistical analysis to confirm that there is some differences. During all this project, we have followed that procedure. We always start with detection in order to see if there are some residues or some microbes are present on open surface or closed system. When we see something, we will characterize those flora via the metagenetic. On those areas where biofilm or organic residues will be confirmed, we will apply an enzymatic cleaning and curative treatment in order to get rid of those germs that are inside a protective layer. And at the end of the study, we will explain how we can prevent this load of organics to be present again in open surface or closed systems. Regarding detection, we have used two different techniques. The first one is the biofilm detection kit. These tools allows us to locate some organic residues or biofilm residues that can be present in open surface. You can see here some pictures with the blue dyeing solution that is trapped on organic residues or biofilm. Thanks to that tools, we can locate failure of cleaning procedure in order to see if there is some flora that is present on those specific areas. The second tool that we have used is the UV lamp, because UV lamp can react with organic residue or biofilm and microbes that can be present inside the biofilm. You can see on the video here that when we use the UV light, we can see some areas where there are some residues, and if we swap those areas, we can extract some flora from open surface. This was for detection. In order to characterize and see exactly what kind of germs we have on open surface or closed system, but also on the finished product, we have used the DNA analysis. The goal of this technique is to extract DNA from all microorganisms that can be present. And after that, we analyze those DNA sequence and we translate into flora, the flora that is present on the open surface, on the environment, but also on the finished product. In order to treat biofilm or organic residues, we have integrated specific enzymatic cleaners, and those cleaners can be active on organic residues. We can say tenacious organic residues or biofilm structure. These are two different enzymatic cocktails that we have introduced in order to guarantee the complete extraction of all residues from open surface. So we have used two different products. First one is the Biorem foam. It's a foaming product for open surface. The second one is the Biorem CIP. This product is designed for closed system treatment in order to fully act on tenacious organic residues and biofilm structures. So in order to prevent biofilm to regrow again in open surface or in production premises, we are combining the use of curative solution. Here it's the red line. So when the biofilm is present in an open surface, thanks to the different tools to detect that biofilm, we can act with that curative treatment. And after that, we are using a preventive solution in order to avoid this biofilm to regrow in the equipment or in pipes and open surface. So it's the combination of curative treatment first, and after that, some prevention thanks to some specific enzymatic product. So this was the global approach that we have used in this research program. Here you have the results of those of that analysis in that specific application. So this graph gives you the results of the total flora concentration on the finished product, depending on the different kind of cleaning procedure. You can see here dark blue, so it's the standard and conventional cleaning. So it means alkaline-based product and sanitation. Here, dark green is the curative treatment, so reinforced enzymatic cleaner. And after that, you have the pale green. So light green, it's the enzymatic treatment 3 times a week, 2 times a week, and 1 time a week. After that, we restart with conventional cleaning, and we applied again a curative treatment. You can see that on average, so the 50% of all analyses are going down when we introduce the enzymatic cleaning. When we get back to conventional cleaner, we increase the amount of germs in the finished product. And when we get back to the enzymatic treatment, you see that we have a reduce of the amount of the global amount of germs in the finished product. So here we can confirm, thanks to statistic tools, we can confirm that the use of enzymatic product is helping to reduce the amount of germs, the total flora, in finished product at the end of the process. So this is the first conclusion of this analysis, is the enzymes helps to reduce the amount of flora, the global amount of bacteria in the finished product. Second information, thanks to the metagenetic analysis, we can compare and we can analyze the type of bacteria that are present in the finished product. In green, we have those bacteria that are more from the technological bacteria, or it's those germs that are not altering the finished product. And in red, you have those spoilers. So those bacteria or microorganisms that have an impact on the finished product. You always have D0, so it's at the end of the production process, at the end of the shelf life, and 7 days after the shelf life. So before doing any enzymatic cleaning, you can see that you have a massive increase of the spoilers in the finished product when day after day. So at the end of the shelf life, we can say we have 1/3 of non-altering bacteria and 2/3 of altering bacteria. You can see that when we apply the enzymatic cleaners, we are reducing the amount of spoilers on finished product on D0, so at the end of the production, but also at the end of the shelf life, and also at the end of the extended shelf life. So it means that here, these are the best values. Is if you apply a curative treatment followed by at minimum 2 enzymatic cleaning per week in that kind of application, we have a very low load of spoilers at the end of the production time, and at the end of the shelf life, you still have a low load of spoilers, and it gives us the opportunity to extend shelf life of the finished product. So thanks to the statistical tools, in this case, we have used Mann and Watney process, we confirm that there is a real difference between conventional cleaning and enzymatic cleaning. And there is a real impact at the end of the production, at the end of the shelf life, and also at the end of the shelf life plus 7 days, so plus 1 week in this case. Quite interesting conclusions. So, conclusion of this study is to say we achieve extending the expiration date or the shelf life of the food products by doubling, in this case, the estimated shelf life. Another example coming from this study is on the fresh hamburgers. You can see here that this is the shelf life with standard cleaners, alkaline-based product followed by a sanitation. This is the shelf life of the fresh hamburgers with after a curative enzymatic treatment. And if we apply a curative treatment followed by a preventive treatment, enzymatic treatment, we can extend shelf life up to 24 days compared to the initial 11 days. So again, in this case, we are able to double shelf life of the finished product. And in this case, it's fresh hamburgers. So it's quite interesting if we want to avoid waste of any product that are not able to be consumed anymore. So the efficiency of our green chemistry that is based on enzymes first allows to extend the committed shelf life of food products. And obviously, it reduces the food waste on all levels of the food chain. We proved that losing control of surface hygiene can be linked to the presence of biofilm. And on those samples we collect from the food areas, food premises, we analyze open surface samples. And on those samples, we have identified many bacteria that are able to produce biofilm. So they are well known to be biofilm formers like Pseudomonas, for example. We highlight a lot of Pseudomonas from samples we collect from open surface in the different application we followed in this project. So some information about biofilm. First, biofilm is a constant risk of contamination. Inside biofilm, you have bacteria, and you have those bacteria that are producing protective matrix. This protective matrix is organic. It's organic polymers like polysaccharides, proteins, DNA, etc. Thanks to that protective layer, those germs are quite resistant to any sanitizer and biocide that you can apply on those bacteria. It's well known that more than 80% of the microbial biomass on our planet is organized into biofilm. 60% of foodborne illness are caused by biofilm, and we know that all those germs are able to produce biofilm. And a study also have demonstrated that if a microorganism is found in the environment, you have more than 70% of chance to get this microorganism in the finished product. This is the biofilm growth cycle. It always starts with some planktonic cells that are present in the environment. It can be in the air, it can be in the raw material, or it can be also in the operators. So those bacteria will enter into the process. They will mechanically attach on surfaces, and they will start to produce this protective matrix. This biofilm is going to grow. It's the maturing phase. And when the biofilm will be mature, it's going to release some part of the biofilm. It can be planktonic cells, or it can be also fragment of biofilm. And it goes back to the initial phase, and you will get some other contamination in other location. So this is the biofilm growth cycle. Many bacteria or microorganisms are able to produce biofilm. And in this study, we have mainly analyzed, we have highlighted Pseudomonas and Leuconostoc, for example, in this case. But you can see that you have other germs that are present in the food industry or in the kitchen areas, and they are capable to produce biofilm. Even some Candida albicans, for example, is capable to produce biofilm. Inside that biofilm, you can have many other microorganisms like viruses or phages and yeast and molds. They will be happy to live in the protection of the biofilm produced by those initial bacteria. There are many scientific studies that are giving some additional information about biofilm and this, the specific capacity of biofilm to resist to any sanitizer. And in this case, for example, you can see that they have applied high concentration of active chlorine. So in this case, it's 1000 ppm of active chlorine for 20 minutes. And you can see that it's not fully damaging biofilm produced by Listeria, Staphylococcus, or Pseudomonas. So it's important to understand that it's not bacteria themselves that resist to sanitizer. It's because they are inside a protective layer. And if we take that planktonic cells and just with 10 ppm of active chlorine for 30 seconds, you are killing the bacteria, where they have demonstrated that even 1000 ppm for 20 minutes is not fully effective. A second article, a scientific article that is quite interesting, is they're talking about the viable but not culturable states of pathogenic bacteria. It means that those bacteria, when they are inside the biofilm, this biofilm stress can induce this VBNC mode to some pathogenic bacteria. So it means that if you have those bacteria in your food equipment and you put them into a growth media, maybe that bacteria is not going to grow, is not going to develop into a colony, so you will not be able to see and to identify that contamination. So it's a reason, for example, in some case where you have some complaints from the market, but you don't see any impact or any, you never saw any issues in your quality control. Maybe it's because you have some VBNC state bacteria in your process and you don't highlight those contaminations. In conclusion, the conclusion of this study is to say that there is a significant reduction of the amount of the total flora concentration in the finished product if you compare enzymatic cleaning and standard conventional cleaning. So thanks to the enzymatic solution, it's possible to reduce the amount of bacteria in the finished product at the end of the production process. Also, we know that we have extract some microorganisms from the equipment thanks to the enzymatic treatment. And some part of some equipments were highly contaminated. So we can understand that if you have some resistant bacteria that are present in the processing equipment, you can have a transfer from the processing equipment to the finished product. And you are increasing the amount of bacteria at the end of the process. We understand also, thanks to that study, that you need to apply a minimum frequency of enzymatic cleaning to prevent contamination to be present again in the food facilities. And one main conclusion is to say that the microbiological quality of finished product can be improved thanks to enzymatic cleaners. So this is the end of this presentation. So if you have any questions, now feel free to use the box Q&R to ask your question, and I will try to answer all those questions right now. You will obviously get that presentation by email at the end of the week. So if you have any question, do not hesitate to answer that email that will be sent by the end of this week. Okay, so I leave you the mic. You can ask any question you want. Nobody there to ask any questions? It's always like that, so we have to wait a few minutes for people to type the questions. Okay, so far no questions. I'll leave you a few seconds. Okay, so as conclusion, one question is coming. No? Okay, just someone says that it's good presentation. Thank you. It's also, it's not a question, but yes, it's important to say. Thank you very much. So in conclusion, we can say that the efficiency of our green chemistry based on enzymatic solution allows to extend shelf life of food products and also can impact and reduce food waste on all levels of the food chain. It can be the case in food industry, but also in restaurants and also in a different other application. It's proved also that losing control of surface hygiene can be linked to the presence of biofilm. So it's quite important to use some specific tools to detect potential biofilm contamination in order to manage and to monitor and avoid this biofilm to be present in open and closed system, in order to avoid any transfer of germs that are present in the biofilm in the equipment process equipment to the finished product. And I think we have a question. Okay. So what about old and young biofilm? Same dose, same treatment. Okay. Thank you, Pablo, for this nice question. And hi, Pablo. No, fresh biofilm is more easy to remove compared to old biofilm. That's why in our different procedure, we have a procedure for old biofilm, so it's a curative treatment. And we have a specific process for young biofilm. So the idea, we always start with a curative treatment to get rid of old and very huge biofilm. And after that, we can prevent biofilm with a second solution in order to treat those young biofilms. And young biofilms, because young biofilm are less strong, less resistant to old biofilm, because in old biofilm, you applied a lot of aggressive solution and it makes organic residues more resistant to conventional and enzymatic cleaning as well. So that's why old biofilm, we recommend a curative treatment, the reinforced solution, and for young biofilm, a preventive treatment must be fully effective. Okay. So I leave you a few seconds maybe to ask another question. Okay, there we are. So I see on the list that many companies join from different locations. So it's quite interesting to see that you are all present. And I hope you like that presentation. And if you have any question, feel free to ask. We will be happy to answer your questions. Hope to see you soon. Take care and have a nice afternoon.